When you see the genetic code table, does it look like alphabet soup? Well, I can help. There are three learning objectives. Number one, we are going to learn how to read and interpret the genetic code table. Number two, we will prepare sequences for use with the table. And then number three, we'll work through questions and do step-by-step solutions so that you get a better understanding for how to solve these questions. This is three questions solved, and I am Dr. Carmel Rogers. Make sure to have something to write with, and let's jump in. You may remember from last time we talked about the central dogma of molecular biology. And the central dogma goes over how information flows within the cell. Here we have a cell. In purple is the nucleus. Notice how the DNA is inside of the nucleus. DNA is double-stranded. And DNA, you can think of that as the blueprint of our bodies. So DNA gives instructions to our cells in order to make what we see in the mirror. Now, the process of the central dogma, we start off with our double-stranded DNA, and then we make a copy, and this is called the messenger RNA. Notice how the mRNA is single-stranded. So the process of going from DNA to RNA is transcription. The enzyme that does this is RNA polymerase. Now, the next step is translation, and that's just going from the mRNA to protein. And the mRNA is able to leave the nucleus and go into the cytoplasm where it joins up with a ribosome. And then the ribosome does a lot of the translation. It's able to do what we're going to do today. It looks at the codons on that mRNA and it will associate with a transfer RNA, which will add in the correct amino acid sequence. I have questions here, and we'll just start with number one, and I'll show you how to use the genetic code table. In the last video, we worked on the different strands, the different nucleic acid strands, whether it was coding or template, the messenger RNA or the tRNA. And you'll need to understand those associations as you work through these types of problems. Here it says identify the amino acid sequence if the messenger RNA sequence is shown. Now, if we have the amino acid sequence, you can think of that as relating to the messenger RNA. Within the genetic code table, these are codons. And with the messenger RNA, we're going to want it in 5' on the left and then 3' direction on the right, and then to use the messenger RNA. So the way that we read this, the first letter is on the left side. The second letter is at the top. And then we can find the third letter on the right side. Let's go ahead and look at our sequence. Now, we can use this one directly because the mRNA sequence is shown in the 5' to 3' direction. If it started off with 3' instead of 5', then we would need to do some changes to put it in a direction or orientation that we can use in the table. But since it's already in the 5' to 3' direction, that's what we need. So we can use this as it is written. So we have the first codon, which is CAU. We'll look for C in the first position, then A in the second position. We go to where these two intersect. So it's somewhere within this square. And then the U is in that third position. So we can find the U at the top. So here's our CAU, and it codes for the amino acids histidine. Our next one is CGU, and we're just going to use the same process. C in the first position, G in the second, and then we're going to find the U. And this one codes for arginine. Our last one is GGA, G in that first position. Also G in the second position, and then A. That will be glycine. All right. Our answer is B. Let's try the next one. Identify the amino acid sequence if the coding sequence is shown. Now, we talked about the acronym Catchy, Trendy, Music Time. And this will help us to understand the associations of the different strands. Since they didn't give us mRNA, we need to convert the coding sequence to the mRNA sequence, and then we can use our genetic code table, just making sure that we have, again, the mRNA with 5' on the left and then 3' on the right. So we can go ahead and use our acronym coding template, mRNA, tRNA. So coding going to mRNA, it skips a level. That means that this association is identical. But since mRNA is a type of RNA, it's not going to have any T's. It's going to have U instead. So let's go ahead and write our mRNA sequence. And again, it's an identical comparison. So we have 5', the G's are the same, the A's are the same. Instead of a T, we have a U. The C's and the G's are the same. The C is the same. The A is the same. Instead of a T, we have a U. Okay, so that's how we go from the coding sequence to the messenger RNA. And now that we have the messenger RNA, we can use the genetic code table just like we did in question number 1. We're looking at GAA for the first codon. So here's G in the first position. Then we have our A. And then we look for A in the third position. So this will be glutamic acid. Our next one. Those aha moments are so special. Let me know in the chat if you've had an aha moment during this session. And also like, share, and subscribe. That will help me to grow my channel. Let's get back to the problem. And I'll just put A period A to make our amino acid sequence. Our next codon is UCG. And we'll just go back to the table. U in the first position, C in the second position, and then a G. That is serine. And our last codon is CAU. Here's the C, here's the A, and then we have the U. This would be histidine. So our answer is B. All right, how are you doing so far? Feel free to pause if you need to. You can also rewind. Let's go to our last question. What is the amino acid sequence that results from this template strand? So we're going to have to use our method for changing strands. If we have our template strand, and again, remember we need the messenger RNA, 5' on the left and 3' on the right. We'll write our acronym, catchy, trendy, music, time, and we're going from the template to the mRNA. They're just one level away from each other, so that means the association is complementary. With complementary strands, they are anti-parallel. Our original strand starts with 5' for the template, and we want to go ahead and do 3' to start off our mRNA. And again, RNA does not have thymine, it has uracil. So A will pair up with U, T with A, C with G, T with A with U, G with C, G with C, C with G, G with C, and then 5'. Now notice how the mRNA has 3' on the left. In order to use the table, we have to have the 5' in on the left, and every sequence is equal to itself when you write it backwards, as long as it's consecutive. And the way that we do that, we'll just put 5' and we'll work our way backwards from the 5' end to the 3' end. So 5', then C, G, C, C, U, A, G, A, U, and then 3'. Okay, so this is the sequence that we'll use. It's in the correct orientation and it's mRNA. The first codon is CGC. Here's our C, here's our G, and we find it right here. It's arginine. The next one is CUA. C, U, and A. That's leucine. And then we have GAU. G, A, and U. That's aspartic acid. So our answer is C. Now notice this last option. It says there is a stop codon, therefore no amino acids are made. We are going to talk about point mutations, and one type of point mutation is a nonsense mutation. So make sure you check out that video. I just want to give you a quick peek. If you look at the genetic code table, there are some that don't code for um, amino acids. They have stops instead. So UAA, UAG, and UGA all code for stops. And once that stop is seen by the ribosome, then translation no longer happens. And so, as we get into the different types of mutations, you might come across questions and you'll see the incorporation of a stop codon. So now you know what that means. Thank you so much for watching me today. Please remember to like, share, and subscribe. And I look forward to you joining me in the next video. Take care and be blessed. I hope you learned something new today. Let's get closer to your educational goals. We can do that by a one-on-one session. I also do group sessions. Check below for my details. I look forward to joining up with you. And please remember to like, share, and subscribe. That helps me to grow my channel. Bye.